Synergistic novel triple drug anti-malarial dry syrup of nanosized atovaquone in vitro dissolution and pharmacokinetic studies. Malaria still remains a major health problem worldwide, and according to WHO data of 2019, about 229 million cases of malaria were reported worldwide. There is an urgent need to devise strategies to reduce the disease burden. The five artemisinin combination therapies recommended by WHO are highly effective and well tolerated. But poor absorption of the drugs, high dose regimens, need for food intake with the medicines, patient non compliance, and complicated regimes, along with development of resistance to the drugs, are some limitations associated with the current therapy. Thus, a new combinatorial antimalarial treatment regimen is the need of the hour to preserve the efficacy of especially artemisinin derivatives. It has been reported that mass treatment with ACTs alone is unlikely to be sufficient for malaria elimination, and hence primaquine and or atovaquone proguanil may be worth adding. Malarone tablets containing atovaquone proguanil show variability in absorption with only 3% of the drug dissolving in simulated gastric fluid. It has a good safety profile, but the high cost of atovaquone and the influence of food on absorption limits its use. The product is essentially unaffordable in malaria endemic areas, especially in the third world, and it is not sold in India. The drug needs to be processed to improve its aqueous solubility for enhanced absorption. Therefore, the first objective of the study was to fabricate a nano-sized atovaquone by a green approach for dose reduction and to enhance dissolution and subsequently its bioavailability. There is no artemisinin combination therapy available which acts on both liver and blood stages of the parasite for better control of malaria. Many triple drug artemisinin combination therapies have undergone trials, and only artesunate atovaquone proguanil combination was found to be safe and highly effective in multidrug resistant malaria. But the drawback of this therapy is high dose, high cost of atovaquone, and there is no co-formulation available containing all the three drugs. The main rationale behind addition of artesunate, which has short elimination half-life, this addition of this drug to atovaquone proguanil will rapidly remove the parasites from the blood due to schizonticidal and gametocidal action of artesunate, killing more than 99% of the parasites before the drug gets eliminated. And later, the residual parasites will be cleared by the intermediate acting proguanil and the long acting atovaquone. These partner drugs will help to maintain efficacy till 4 asexual life cycles of Plasmodium parasite to cure the disease completely. All the three drugs kill the parasites in the blood stage. Due to different sites of action, there will be better control of infection. Due to different modes of action and different biochemical targets, the chances of development of resistance to these three drugs and reoccurrence of parasitemia is minimal. Therefore, the second objective of the work was to prepare a novel artemisinin combination therapy dry syrup with synergistic action using nanosized atovaquone with improved dissolution property. Coming to the first part of the research, that is nanosuspension. Nanosuspension is the most preferred formulation system for atovaquone owing to the poor solubility of atovaquone in water and various other solvents, including oils and lipids. It has other limitations of high dose, high log p-value, and high melting point. Therefore, a pH-based novel method, which is simple, organic solvent-free, scalable process, was utilized to fabricate nanosuspension with a particle size below 300 nanometers. This nanosuspension was compared with other nanosuspensions prepared by two other methods, such as top-down method and anti-solvent precipitation method. The nanosuspension was converted into solid form by freeze drying, and by adsorption. The formulations were evaluated for various physicochemical properties. The in vitro dissolution was carried out using USP apparatus 2 with OGD dissolution medium, that is 40% isopropyl alcohol in pH 8 phosphate buffer. But there was no significant difference found between the dissolution profile of nanosuspension and the plain drug suspension. Hence, dissolution was carried out in 500 ml 2% chromophore EL solution, which was employed as the discriminatory medium using the similar apparatus. This medium was selected based on the solubility studies of the drug carried out in various media. The pH-based nanosuspension showed significantly higher dissolution, with the freeze-dried form showing 63% and adsorbed form showing 46.8% dissolution, as compared to the plain drug suspension, which showed only 8.7% dissolution. Solubility studies in water and at different pH revealed the enhanced solubility of the freeze-dried pH-based nanosuspension as compared to the adsorbed nanosuspension, as well as compared to the plain drug and the other nanosuspensions prepared by the other two methods. The in vivo studies, pharmacokinetic studies of the nanosuspension showed twofold increase in AUC and a quicker onset of action as compared to the plain drug suspension. In vivo pharmacodynamic studies were carried out in mice with Peters 4-day suppressive test. In this test, the prophylactic efficacy of nanosized atovaquone was proved at 1/80th the therapeutic dose. In another study, that is clinical simulation study for complete cure, the synergistic potential of artesunate with the nanosized atovaquone and proguanil was proven, leading to a significant dose reduction up to 1/80th the dose of atovaquone proguanil and up to 1/5 the dose of artesunate in the synergistic combination. The nanosized drug was incorporated in a novel triple artemisinin combination dry syrup, which is easy for administration to bedridden as well as pediatric and geriatric patients. This triple drug co-formulation was evaluated for in vitro dissolution using a similar discriminatory medium as mentioned earlier. Both proguanil and artesunate showed 90% dissolution in 15 minutes, and the enhanced dissolution of atovaquone from the dry syrup was 8.8-fold as compared to the unprocessed atovaquone dry syrup. In conclusion, a novel organic solvent-free scalable pH precipitation method was invented for preparing nanosuspension of BCS class 2 drug, that is atovaquone, to increase its dissolution and bioavailability. The potential of the synergistic co-formulation of nanosized atovaquone proguanil along with artesunate was proved in curing the malaria at reduced doses of all the three drugs. This can serve as a solution to the pill burden and multi-drug resistance observed with the current therapy. The product will turn out to be a boon for poor patients suffering from multi-drug resistant malaria in malaria endemic areas of the world, including India. Thank you.